Hello, hello. I'm seeing that Professor Gonzalez is here, so we are almost ready to start. As you know, we were gonna stream live in the Facebook of the university, and so I'm gonna wait for her to set it up, and then we managed to start. Okay. Today we're gonna have a talk about bioinformatics. So technology students, chemistry students mainly, but I want to say that also engineers are going to be pretty interested in that. And we will see the research that Dr. Hegel have done during one of her postdoctoral research fellowship. So just 2 minutes and
We are live now.
I think we are live now. Fantastic. So we can start. Hello, everybody. Welcome to our third day of the STEM week. I don't know which number of talk we are now. I think we are around fifth talk, maybe. Today we are taking advantage of this virtual edition of STEM week, and we have our host streaming from Barcelona in Spain. She is Dr. Samira Jaeger. She is a bioinformatician by training. She started her career in Berlin to the Freie Universität of Berlin, and she done her bachelor and master degree in bioinformatics. During her studies, she had opportunity also to go to do two internship, one at the Center of Molecular Medicine in Berlin, and the other one in the Monash University in Melbourne, Australia. She worked after the master degree in the IBI in Cambridge as a visiting scientist. And then she finally started her PhD at the Humboldt Universität of Berlin in Germany. Finishing her PhD, she moved to Barcelona for her postdoctoral research fellowship at the Institut de Recerca Biomédica, IRB of Barcelona, where I had the opportunity to finally meet her. And now she is currently working for Hotspot Therapeutics that is based in Boston. So she's not new to these online conferences. Today she will speak about her research, and the title of her talk is Quantification of Pathway Crosstalk Reveals Novel Synergetic Drug Combination for Breast Cancer. I give you the word and the space. If you want to stop asking question or waiting for question, guys, you know that you can send chat messages. You can even open your microphone and just ask questions. And happy to have you here, Samira.
Yeah, thank you, Federica, for the invitation, and thank you for the opportunity to present my research to you. Yeah, so I will jump in directly. So I guess you can see my screen well. Everything is fine. Yeah. If you have questions, I think I will not see the set here. Like there's too many options with the Zoom. So maybe just if there's something not clear during the talk or you have some questions, so we can just, so it's less formal, we can just chat about it. So just let me know. Otherwise, we talk about anything that is open in the end. So yeah, as already introduced, what I want to talk about today is a pathway crosstalk quantification and how this can be used to reveal synergistic drug combination for breast cancer. And as already said, this is a project that I like very much that I did during my postdoc at the IRB in Barcelona in the structural bioinformatics and network biology group. So as we all know, cancer belongs to one of the major health burdens of our society, together with heart diseases, diabetes, and respiratory failure. And despite the advances in the last year in finding new agents and treating tumors better and better, one of the major problems or the challenge that still remains is basically treatment failure. So basically the cancer and the tumor cells, they find a way to evolve, and basically they find a way to evolve into compensatory mechanisms that can antagonize the therapeutic effects. What does it actually mean? That after a treatment, after some prolonged treatment, the drug become inactive. There can be a loss of target expression. So imagine there is like for instance, in the HER2 positive breast cancer, that is basically characteristic for a receptor that is overexpressed. If suddenly this receptor is not expressed anymore, basically the drug becomes inactive, or any other mutations of the drug targets themselves, or activating mutations downstream of the intended drug targets. Then we can have modification of target regulators and the establishment of alternative signaling routes through pathway crosstalk. So we can, this whole process, we can envision like this. So imagine this is a set of tumor cells where we will basically, that we subject to a certain treatment. Some may have some mutations from the beginning. We give our treatment, and then we see that the tumor cells, they die. So basically the tumor shrinks, and and basically there's a good prognosis for survival and so on. But then what happens very often is that all these mechanisms that I just mentioned, that basically they lead to a tumor relapse, because even though that most of the tumor cells have been killed through the treatment, at some point, basically these ones that are resistant to the therapy that has been given, they start to proliferate again. And basically this is usually when the tumor reoccurs. And so basically a tumor relapse and the treatment fails, basically due to a drug resistance. To give a little bit more, let's say, visual explanation. So this is a simplified schematic of the signaling pathways driving graft resistance to the BRAF inhibitors. So the BRAF inhibitors, basically, BRAF has this characteristic mutations, the B600E, where basically drugs have been successful developed to inhibit this mutation. And this mutation is dominant in melanoma. And basically these drugs make melanoma, treat melanoma very well. Basically what it does is that cells are sensitive or highly dependent to this BRAF protein, because it activates the MAP signaling pathway and leads to proliferation, cell proliferation, and survival. So if we stop this BRAF protein, basically we stop the MAP kinase signaling, and we are stopping proliferation and also the survival of the tumor cells. But what often happens after following a chronic BRAF inhibition, the cells become resistant and evolve an array of compensatory mechanisms. So the mechanism that I just mentioned. One of them is, for instance, that other RAF isoforms are switching and being activated, and they start to send signal into the MAP kinase signaling pathway, and basically circumvent the initial inhibition of the BRAF protein. There's other receptor tyrosine kinases that might be activated also in this process, as well as the engagement of the PI3K pathways that can be done. That also promotes survival, and which is also favored by a mutation in the tumor suppressor gene, the PTEN. So we can see that there's several points basically where the cell evolution or tumor cell evolution finds the way to basically circumvent the initial inhibition. And understanding the signaling network that drive drug resistance enable us to develop rational drug combinations to target melanomas that are refractory to BRAF inhibitors. So that means, for instance, we could inhibit these active receptor kinases with different inhibitors. We could inhibit the PI3K kinase. We could inhibit the MAP kinase pathway directly, or we could inhibit the ACT. So the idea is basically to find which are the best combinations that avoid this pathway, this crosstalk between the pathway, and which basically avoids a signaling that leads to the resistance of in this case the BRAF inhibitor, but of any inhibitor in general. So I don't know how familiar you are with biological pathway. Now you have seen one, but biological pathway in general, they are a series of interaction among the molecules of the cell. So let's assume proteins that lead to a certain product or change in the cell. So imagine you have a signal and that is being produced in your body. Basically, this activates a certain protein, and this initiates biochemical reactions that can lead, for instance, to cell proliferation or apoptosis or cell survival, all the biological functions that we basically know. It can mean that, for instance, a protein becomes activated through phosphorylation or becomes inhibited through dephosphorylation and so on. So all the biochemical mechanisms that we can think of are basically interlaced, and they all go to a common goal, basically. Yeah, so as I said, such a pathway can trigger the assembly of new molecules, such as fat or protein molecules, turn on transcription of genes, or turn it off, or prompt the cell to move, amongst many others. And ideally, when we think about biological pathways, people often think about the linear chain of the biochemical reaction, that is going from the bottom to the top. But mostly they are very complex networks that involve certain feed-forward and feedback loops, better. So they are not this linear chain. So just to give you an idea, this is the apoptosis pathway that is basically stored in the KEC database, and we can see basically, so we have input proteins here, basically signals, and then we have apoptosis, basically as the main output of this pathway. But then we can see that some go basically in the right direction, some errors, some go up, and some even go back. So these are the feedback loops basically that I was just mentioning. So it's not a linear chain. First protein A, then protein B, then protein C, but basically they are all interconnected, and just changing a little bit in the equilibrium of the cell might lead basically to a loop or any interaction with other protein partners. So this is basically like this is the motivation for or basically one of the explanations for drug resistance. And basically, as we know, so one of the most promising therapeutic strategies for better clinical outcome in complex diseases is combinatorial therapy. And this has been very well proven in HIV, that now HIV is very, let's say, very treatable, giving a cocktail of, I think at the moment they're giving 2 or 3 different drugs, and basically by this they're people that have acquired this disease can live almost disease-free for a much longer time than before. And also, since a couple of years, basically combinatorial therapy is the new, or not the new, but is a strategy that is used as well in cancer. And that basically means that we want to clinically intervene at different at the level of the distinct adaptive response mechanisms that are related to treatment failure. And what we expect from this is not only greater clinical efficacy, while overcoming toxicity and adverse effects that are commonly associated with higher drugs. So one simple solution for cancer is we just give a lot of drug, but we know that all those chemotherapeutics, they are very, very strong on the body. They have very big toxicity. So this is not the solution. But we hope that by combining drugs with different mechanisms, not only are they working better, so they have a greater efficacy, but also we can use smaller doses at the same or a better result that are much less harmless for the patients, basically. As we know how hard therapy can be on the patients. So one of the problem at the moment, or when I did the study, is that most of the approved combination resulted from empirical clinical experiments. So patients go into treatment failure, for instance, and then the doctor think about, combinations that they could try to help this patient to live a couple of months later. And then they do studies in smaller, let's say in smaller clinical trials, and combinations that work are being given more frequently. But given the large amount of drugs that are out there, actually, it is not only the drugs, but also the different cancer types. It is not feasible at the moment. I mean, more and more techniques are generated, but do all the pairwise combinations of drug combinations to see which drug combination could be as efficient. And basically, this approved drug combination that result from empirical clinical experiments, they lead to several Me Too. Yeah, I think there's also called Me Too combinations, meaning that if we have a drug combination including two different drug classes or drug family, then if one works, researchers or doctors are much more inclined, okay, let's use another drug from the same drug family, let's say. And we can see, for instance, here in this plot, I plotted the different, let's say, drug families. So we have CDK inhibitors, EGFR inhibitor, heat protein inhibitors, microtubule modulators, and so on. And basically, the larger the circle, the more therapies exist having one of the members of this drug class. And we can see that there are some drug classes that are frequently combined, and then there are some drug classes that are not, or they have not been tested, or they have shown to be not relevant. So this is basically something that we want to circumvent. And as the title of my talk says, that we didn't try to tackle all the mechanisms, but we wanted to look at alternative signaling routes through pathway crosstalk. So that was the focus of our study. What do I want to talk about today is basically about what is pathway crosstalk and inhibition, and how we can quantify it, then how we apply this and apply and evaluate it to common drug combinations, and then the experimental validation of selected drug combinations in cancer cell lines and a mouse model. So basically, experimental studies have demonstrated that cancer cells can adapt the signaling pathway circuits upon treatment, and basically can establish alternative signaling routes. So this is basically a therapeutic signaling network for the estrogen receptor, of course simplified, and we see that estrogen is coming, is binding to the estrogen receptor, and the result is basically proliferation. If we inhibit the estrogen receptor, basically proliferation is inhibited and the tumor cannot grow further. But then most of the pathways are interlinked. So in this case, we have the red pathway, the VEGFR pathway, and that is basically, we can see that gives also some input to some of the proteins of our estrogen receptor pathway. So what happens basically, so we have one signaling route we have inhibited, but basically, or there is from the beginning a second one, or this could be, like in the earlier example, that basically a signaling route that is being established. And what basically happens is that the proliferation is being the inhibition of the proliferation is being circumvented. So it's not, proliferation is not inhibited anymore because we are receiving, the pathway receives information from the other related pathway, and basically our therapy or our drug will fail. So for this reason, we try to identify pathway crosstalk between breast cancer drugs. So in this case, we focus on breast cancer, but this can be applied to any other cancer type as well. And what does pathway crosstalk mean? It means basically proteins and interactions, or that are basically shared between two pathways, for instance. So we can just overlap these. We take basically pathways as defined in the KEC database, and then we can overlay them, and we can determine which are the proteins that are shared between two pathways. And then basically the idea is to determine the crosstalk inhibition through individual or combinatorial perturbation using network efficiency. And basically by measuring the flow information through the crosstalk, So basically what we want to do is how much, so the question is how much of the pathway crosstalk can we abolish when we use one drug or the other drug or the two drugs together, basically. And for this reason, for this purpose, we use the network efficiency measure, which is a topology-based measure to determine the flow of information within the network. So in the network biology area, there's several centrality-based measures that can basically tell us about the importance of a node. And in this case, we use this measure. Just to give you an idea, it is defined as the sum of inverse lengths of the shortest path between all the network elements and can be computed by this formula. What is more important to know that network efficiency ranges between 0 and 1, where 0 means all proteins communicate directly with each other, and so that it's a fully connected network, and so that means 1, and 0 means there's no flow of information in the network. So if we look at the 3-toy network, the blue one would be network efficiency of 1 because all the proteins are directly connected and they can basically the flow of information is just one step away. And then in comparison, basically this, where we have no interactions, there's no flow of information, so the network efficiency would be 0. And then here we can see that the proteins are connected with each other, but not all of them. So for instance, to come from this protein to this one, so the shortest distance is 2. And so we need a longer time, basically, or we need more time to send information in this network. And this is basically the basic idea. And then what we're doing is basically we compare the network efficiency before and after inhibition. So we know, okay, we have so much of the crosstalk, and then if we apply one drug or the other drug, how much crosstalk can we inhibit? And probably this becomes a bit more clear with this example. So this is the pathway crosstalk that we have identified, and the network efficiency is 0.29. And we can basically see, and if we use the drug 1, the crosstalk inhibition is 0 because none of the crosstalk is being affected by this drug. If we now test the second drug, we can see that some of the crosstalk is being affected by the second drug. So the network efficiency is only 0.05, and the crosstalk inhibition for the second drug would be 0.83. So we are abolishing, or we are kind of taking out several edges for the flow of information. And then that would be the ideal case. We are applying the two drugs, and the network efficiency is 0, and basically the crosstalk inhibition of this drug combination is 1, meaning none of the crosstalk persists. And we did a couple of benchmarks. Maybe for the time being, I will skip this. And so basically what we did, we took 64 different breast cancer drugs that were in the databases at that time. And then we looked at the current drug combinations for breast cancers that were either approved or in clinical trial. And then we basically computed the pathway crosstalk for all of the breast cancer drug combinations. And basically this is visualized here. So on the y-axis, you can see basically the different drug families. So meaning here, for instance, in red, these are all microtubule inhibitors and modulators. And so the color means if it's white, there's no pathway inhibition, no crosstalk inhibition. And if it's dark green, there is a lot of pathway crosstalk inhibition. And then I also marked basically the drug combinations that are currently in use and the drug combinations that we later selected for evaluation. So we can see that here, this is something that we saw also in the other figure. There's a lot of drug combinations with the microtubule modulators. But then the rest of the matrix, let's say, is quite poorly covered from a clinical point of view. Basically, from this, we then decided to select 10 drug combinations, and we tested them in the human cancer cell lines and 1 non-transformed breast epithelial cell lines. And we selected this drug combination in a way that we didn't want to test combinations from already approved drug families, because the the likelihood that they are working is much higher than for unknown combinations. And basically, so this just gives us an idea about the breast cancer cell lines. It's a triple negative. This is the estrogen positive. This is another cell breast cancer cell line with the TP53 mutations, and then the PT474 cell line. This is a non-tumorigenic cell line, and then we used also an osteosarcoma cell line. And basically, the quantification of the cytotoxic effects, so how much are the cells affected, has been measured by an MTT assay. And basically, this is a biochemical reactions, and the more viable the cells are, the more purple is basically the readout, and the less viable the cells are, the less purple or whitish, let's say, is the readout. And then basically, so we tested different doses of these drug combinations, and then when we wanted to compute the drug combination index. So the drug combination index is defined as basically comparing the effective dose for inhibiting 50% of the cell viability, so the IC50 value of the single agents with their doses in combination. So this is shown here that we have drug 1 and drug 2, and these are the dose response curves for both drugs. And then in black, we see the dose response curve for the combination. And we can already see that the IC50 value for the combination is much lower than for the two drugs together. And then using this drug combination index, we can basically define our combination as being synergistic. This is what we are aiming for, additive, or antagonistic. So what does that exactly mean? Synergistic, additive, and antagonistic? It means that an antagonistic drug combination is the combination of drugs leads to a smaller effect than expected, and we need a higher drug concentration to reach the effect of the individual drugs, right? So we are comparing the IC50 of the individual drugs with the IC50 of the drug combination. It means we need to put much more drug combination to reach the same effect. So this is something that we don't want. Additive drug combinations are combinations of drugs that lead to the same effect that you would expect when adding the effect of each drug independently. So imagine that we have two drugs, and basically we are taking half of the concentration of each drug, and we would get the same IC50 as when using the drug combination. So we are not doing much better, but this could be, anyway, it could be beneficial because we are not using just one drug to treat the cancer, but we are using two different drugs, and we would assume that the toxicity is not much higher than using the individual drug. But then what we are looking for are synergistic drug combinations. So the combination of the drugs leads to a larger therapeutic effect, and then the sum of the effects independently caused by the individual components. This means when we combine two different agents, we need less concentration to reach actually the IC50 than when applying the individual drugs. And basically, so we tested 10 different drug combinations. So they are shown on the Y level, and on the X we see the different cell lines, and we calculated the drug combination index, and the index is shown here as numbers, but is also color coded. Green means synergy, and red means antagonistic drug combinations. And we can see that there's some that we couldn't calculate the drug combination index, and in some cases where it's 0, it means that the single drugs didn't have an effect, but the drug combination does have an effect, right? So an individual drug doesn't help, but the combination, yes, it can help. So this is basically the distribution of this index, and we can see that in some cases we are not doing very well because the drug is very antagonistic, but then we are in the area of additivity as well as synergistic. And basically we have, when we quantify this, we have 35% of synergistic drug combination, 38% of additive drug combination, and 12% of antagonistic drug combination. And we compare this with synergistic drug that's identified in experimental high-throughput studies. This ranges between 4% and 90%. So we are already improving basically the identification of synergistic drug combination, which is It is still improvable, but we are already prioritizing drug combinations that could be beneficial for the patients. And basically then we said, okay, now we want to see if this actually works. And we selected one combination for a xenograft mouse model. And we took this drug combination 2, which has so we took into account several factors. It has a good drug combination index, 0.39. It is antagonistic in the non-tumorigenic cell line. It means to affect healthy breast epithelial cells, we actually need to put more drug. So this is also a good safety or a good toxicity assurance that it is likely that normal cells are not being affected in the same measure as tumor cells. This is what we want. We don't want to kill normal cells. We want to kill the tumor cells, right? And then also we looked at the dose reduction index that basically tells us, that compares the IC50 of the individual drug and the IC50 of the drug in the combination. And it tells us how much can we reduce the drug concentration in a combination. And any value above 1 is actually beneficial. So these are like several factors that we took into account basically to select our combination. And then we used atomic immunosuppressed mice. They were 5 weeks old, and basically they weren't. So this was done in collaboration with Angel Nebra's group, also at IRB. And basically they injected MCF7 breast cancer cells directly into the mammary glands. And then the following treatment protocol was used. So first, some estradiol pellets were injected, then tumor cells were injected, and then basically the tumor grew for 1 to 1.5 months. So until it reached a certain size, and once it reached a certain size, basically for 15 days, the mice were treated, and they were treated with the first drug, raloxifene, and the second drug, cabozantinib. So raloxifene is a drug that is already used, is already approved for breast cancer, but cabozantinib not. And then basically every day the mice were treated, and after 15 days of treatment, basically the mice were sacrificed, and then the tumor was extracted. And these are basically the outcomes. Basically we had 9 mice with 2 tumors per condition. In gray, you see the vehicle. So basically this was no treatment. The mice just got an injection, but there was no drug inside. Then here in the middle, we see cabozantinib in blue and raloxifene in reddish, let's say at the different concentrations. Because when we wanted to compare, we wanted to compare not only to the half of the doses that we were using, but also to since in the combination we are using less dose, we wanted to see how this basically compares to the combination. And then basically here we can see how nicely the drug combination works, and how much it is shrinking the normal tumor size. So to untreated, there's a 4.4 reduction, and to just the individual treatments, there is a larger than 60% reduction of the tumors. So this basically shows that this drug combination has indeed a synergistic effect. We also, this is not shown here, we also did so we were weighting the mice, and the mice didn't lose a lot of weight, which is a sign for being very stressed or being very affected by the drug combination, right? So it seems that it was at least as tolerable as the individual drugs. And then we did a little bit more of molecular analysis. So this has been also done by the group of Angel Nebreda. Basically, this is a tunnel staining where we check for apoptosis. So we can see that there's some few points of apoptosis seen in the staining, but when we compare it to the combination, we see that the apoptosis is apoptosis is initiated when the combination is given in a large scale, and then we can quantify this even with statistic. So there's almost no apoptosis in the initial tumor, as such as the vehicle, very little in raloxifene and a little bit in cabozantinib, but then there's a lot of apoptosis maybe initiated for the combination. That is one good sign. And then this is a staining of the tumor tissue for two markers, the famous one KI67, that basically measures, so the protein is more active when there's a lot of proliferation in the tissue. So we see the marker here, it is quite strong. Here we can see the quantification. We see that for raloxifene, the staining is only 30%, so there's less proliferation. For cabozantinib, not so much, but then for the combination, again, the proliferation is going down in the combination. So basically what we are showing is that combination of raloxifene and cabozantinib has a great therapeutic effect in vivo, and there's a dramatic synergistic effect by simultaneously inducting cell death and inhibiting of cell proliferation. And then last but not least, basically we try to generate a simplified crosstalk between the estrogen and the VEGFR signaling pathway for this drug combination. So this is the simplified drug pathway and their crosstalk. You can see they're highly interlinked. And then from this, in red, these are the drug targets. And then we wanted to see whether we can quantify actually which proteins are infected or how much the activity of these proteins are affected. So you see in the colored ones, these are the ones that we did a Western blot analysis, which is shown here. And basically, this is the Western blot of phospho-ACT, phospho-SRC, BCL2, and the cycling D1 with respect to the activity to the pathway crosstalk inhibition. And what we basically can see, cabozantinib has basically no impact on SRC1, but basically where we use the combination, there's no SRC1. So it is basically, this is the protein that receives the input from both drug targets, and it is completely shut down. The same for ACT, BCL2, there's a little bit of activity, and cyclin D1, which is further down here. No, I don't see it. Here, it's really down. So basically with this, we are demonstrating that we are actually, by hitting these two, we are inhibiting proteins and the pathway crosstalk as we basically intended to. And yeah, so this is basically kind of a proof of concept of our method. And yeah, so with this, basically I want to summarize the talk. So I presented to you a network biology strategy to infer synergistic drug combination based on pathway crosstalk. And basically we showed that 7 out of 10 combinations show synergistic behavior in at least one of the breast cancer cell lines. This is a significant enrichment compared to the experimental high-throughput studies, which only yield in between 4 and 90% of synergistic pairs. And then in a mouse model, basically, we can show a dramatic and synergistic effect on the tumor growth, which emphasizes its clinical relevance. And also this approach can be applied to other tumor types and might be also applied to other complex diseases where pathway crosstalk may play an important role. And with this, so basically this talk is based on the publication Quantification of pathway crosstalk reveals novel synergistic drug combinations for breast cancer, where you can basically find more details on the method, on the pathway crosstalk, on the drug combination index. I ran through a little bit, which so there's much more work that I haven't shown, but it gives you all the details that you need to know to understand everything of this work. And with this, basically, I acknowledge my team members from the IRB that basically helped me in conducting this study. Now, since there have been no questions before, if there's any questions, I'm happy to answer them.
Absolutely, yes. Thank you very much, Samira, Dr. Jaeger, for the sorry for the
No worries.
for the talk. It was really, absolutely, really interesting up to me, and it was absolutely great because in my intro to pharmacology course, we just finished up speaking about all the the parameters of toxicity, pharmacodynamic, pharmacokinetic, and it was great that you show those curves response of an inhibitor. So that's an application of what we've seen in class, how we normally, use these parameters, and also the drug combination index. It is there really well shown and really well applied. So also to see how it is the application of all those parameters that are so theoretical, but then you can see it there. So was great. And I learned about this dose reduction index that I never heard about it. It is really interesting also as another parameter to take into consideration.
Mhm.
I have a question about, well, I have three questions actually, but I'm going to start with one. How did you decide to the selected combination of the drugs that were taken up to the cell lines?
That was a bit, it was not random, but it was me, our boss, and then the PhD student that did that the experience. And we were basically, so one thing was basically this image. So we knew which are combinations that have been approved. So we didn't want to test another microtubule modulator with another HERB2 receptor, because the likelihood that this is working is quite high. And basically we were going for completely new drug combinations from drugs or drug classes that have not been tested. And what we took into account, of course, is the drug combination index, the pathway crosstalk inhibition score that we know. So the closer to one, the better, that we have been calculating. And then we did kind of we wanted to test several different drug classes. So we didn't take all the HERB2 combinations. So we took one HERB2, one of these, one of that, right? To have a broad range of combinations. And then everyone had like a list of priorities of drug combinations that we tested. And then basically we did a bit of the overlap and discussed it a little bit. And so that was a bit, it's not random, but we had a little bit of problems when publishing, because publishers wanted to see a correlation of the crosstalk inhibition index and the drug combination index. But since we didn't pick the best pathway crosstalk inhibition combinations, right, because many of them are already in trial or things like this, we couldn't make this correlation. But what we did is this, for instance, I didn't explain it, but we took clinically relevant combinations. We took drug combinations approved on clinical trial and basically computed this for the different combinations. And we can see that this is not fantastic, but there's a statistically significant difference between drug combinations that have a higher crosstalk inhibition index and drug combinations. Random. These are randomly paired drug combinations here on the right side. We just put together. And then these 86 are basically out of the 170. But then there's some drug combinations that interact directly with the DNA, so they have no pathway associated. We couldn't calculate an index for this one, so that's why it's only 86. But we can show that there is a difference basically in the profile, and we can show that there's some relevance for this measure that we were using. And then the second point, since we focused on several, so these cell lines are all different and they're covering different breast cancer subtypes. So we try to give a broad shot to the selection.
Fantastic. Thank you very much. I think that's Dr. Belmonte wanted to have something.
Yes, thank you very much. First of all, I would like to congratulate you about your work. It's amazing. I am really vast about the amount of information that you have created. This is really, really amazing. I really want to congratulate you, Professor. First of all, I would like to see if there's students that have questions before I ask mine. I don't know if anyone of my students or any other questions.
Yes, there are some participants that raised an hand. Jesus, yes, please.
Hello, good morning. My question is, for example, I don't know if I didn't hear correctly, but for example, in this kind of test, how much is the average time that it needs for you to have like concrete results?
That really depends, because I mean, I started to think about a problem. I started to think about combinatorial therapy, and then I started to kind of define an area, because we cannot tackle all the characteristic of drug failure, right? So there's drug inactivation, there's mutations, there's this and that, and basically it took some time that I decided, okay, I want to go for this pathway crosstalk, and then I had to do all the implementation of the measures and also of the pathway crosstalk. And since this is it's a very progressive work, right? So you're stuck at some end, you move a little bit. So honestly, I don't know. I don't remember if now, probably if you have the recipe how to do it now, it wouldn't take me too long to do it. Maybe a month. No, but when I was doing it, I didn't really know where this was going. I didn't really know this is going to be end up in a pathway crosstalk inhibition index. So it's the beauty also of the research. You don't know where you're going to end up. But basically, this part was let's say it took me a year to refine and redefine and all the computational work, but also working on other stuff basically. And then it took another 1 year probably. It took a very long time to do just the drug combination experiments, because we had 5 cell lines, we had 10 drug combinations. Each drug of the combination needed to be tested individually at several doses, and then in combination at several doses, right? So that took quite some hands to do all the work, and then just going to the mice. This is now it takes one and a half months just to grow the mice and the tumor. And then we started out with just I think 3 mice per group. For publishing, this was too little, so we added another 6 mice, and in the end it takes quite some time. But the computational part is much quicker to do, because it basically depends on you, and it's also all these things are quite easy or quite quick to calculate. It's not that you have to run long and extensive and expensive calculations. So that is quite straightforward. It's more how to reach the point that you say, okay, this is something that is cool. This is something that could work. We have to benchmark it, and now that this takes a couple of months.
We have 4 more questions, so maybe let's try to see. So Daniel, please.
Yes, thank you. First of all, I agree with Dr. Velma that it was absolutely amazing. And second of all, I want to ask the relevance of day 15 when trying the treatments. Like, why did you stop at day 15? And can we expect the proliferation to stop decreasing after day 15?
So seeing the results, I would say that it would go further down. So we had 9 mice with tumors per condition, and we had 1, 2, 3, 4, 5, 6 groups per 9 mice that you have to feed, and you have to treat and everything like this. So I think there's no relevance to day 15. We could have gone further to 20 or 30 or whatever, but A, it's going to be expensive. B, the mice are suffering. So it's to find also a balance between not to be like ethically not harmful, right? And I think when we had this data, it shows the trend. It shows basically, I mean, maybe it goes further down the combination, maybe the individual drugs, maybe at some point, maybe 15 days. But when it comes to tumor, for instance, we are not speaking of 15 days. We are speaking about treatments of years, right? You have a cycle of 3 months, then, or maybe a month. But this is something that we cannot do. This is what is done later in the clinical trial. But this is nothing that you can pay and afford, basically, based on mice. And then again, also mice are too different from human to, let's say, to spend so much time, years in treating a mice.
You need also, I think, to take into consideration that it's a model and the life also of a mice, it is not the same amount of longer life as a human. So you can make a comparison, saying 15 days can correspond to a treatment of 1 year in humans. So
that
always
I
you need
don't
to,
know, but yeah, but it's a point. Yeah, they
and also
much.
not to enter inside the toxic index that could come. We want to see the decrease of the shrinking, in this case, of the tumor cells.
Right. I mean, this was our goal. We want to see whether it works. And I think with these 15 days, we worked. So I don't remember, because obviously the mouse work I didn't do, but maybe, so they were like collecting data every 2 or 3 days. And maybe then they saw, okay, this is the trend. This is the data that we are having. This is good enough. We don't need to prolong this much further. So they said, okay, 15 days is enough. I don't know if this is the kind of the rule in oncology, my studies that I don't know.
Thank you very much. We have Fernando Flores, not Fernando Belmont. Sorry, not yet.
Yes, good morning. I was wondering if the same research process could work with the experimental bacteriophage medicine.
Good question. So you were thinking about, but I think maybe this is something that's been done, no? Like combining different antibiotics to bacterial infections. So I think this is being done. And I mean, I don't know if the processes are so similar, and because bacteria are, compared to human tumor cells, are just a small, teeny tiny unit, right? But so I don't know if we can speak about a pathway crosstalk between this within bacteria, but I think something similar might. One needs to look basically at what is going on with bacteria, what is the problem with resistance against antibodies, for instance. But basically, a combinatorial approach is used, but I don't know what is where's this combinatorial approach attacking the resistance against a certain bacteria. So this is something that someone would need to look to verify whether this could be applied. Yeah, so I lack the knowledge on bacteria and antibiotics. But good question.
Thank you very much.
Thank you. Luisa, please.
Hello, can you hear me?
Yes.
Did any mice presented any sort of metastasis or?
No, I think in this case, the study was too short that metastasis could basically occur. So yeah, we just had the primary tumor that was then extracted and was then used for the Western blot and all the staining and stuff like this. I think it takes much longer, even in mice, that metastasis occur. Also, metastasis usually occur when you live for quite some time with a very low tumor burden, but then the tumor cells travel through the body and they basically find a new home in another organ, and there they start to proliferate until a certain point a new tumor can be detected. And in 15 days, I think this is, if you're not using metastasis cells, this is something that you could do. You could just extract cells from a metastasized tumor and inject it into the mice, and then you could see where a metastasis is forming. But in this case, we use primary breast cancer cells, so no metastasis.
Thank you. These are cool results.
And we have Eugenio.
Yes, good morning. I was just asking when you were explaining to my classmate how the process, all this process, which was the most difficult part or most laborious part for you until the entire process. Sorry.
Publishing was quite a pain, unfortunately. It took me very long to publish. I think generating the idea, implementing basically because you're gathering a lot of data, and the data needs to come in a good format. You don't want to compare apples with peers, so you're not like proper data integration and then implementing the algorithms, testing the algorithms. And I think this was the more laboratory, more extensive work that was that took more time to, because then in the end you apply and then you have your results. But if you didn't do a lot of testing before and you did evaluations and benchmarking, you don't really know what this means. And then maybe also you're not only, since we were looking at a lot of new combinations, right? And you try to go into the literature and try to understand, can this make sense? Does this make not to see also if you have to adjust your algorithm a little bit, or if you have to change some parameters or take something else into account. For instance, when you look at it, is a quite basic study. I mean, it's nice, but it's basically so. For instance, in the next step, what I would do, because we are not looking at any gene expression data. So what I would do in the next step to improve this is to look at gene expression data of cancer patients that have a certain subtype, because we know that especially breast cancer, they are different. So, and I think with this you can even more prioritize synergistic or detect synergistic combinations when just looking at the plain pathway data and not taking any gene expression into account. Sorry, now I went a bit off-road.
We have still 3 questions more. Mariano, please.
Yes. These all drugs that you are testing, these are already known drugs, or are drugs that you made with the only purpose of being combined?
No. So basically, what we are starting with was we were starting this with 64 breast cancer agents that you can find, that I was looking up in the FDA Orange Book. So, or either approved drugs for breast cancer or drugs that are in clinical trials. So we didn't create any of our own drugs. We just took also to have a little bit of context and relevance, right? Let's not take, this could be also a next step, just let's take a drug that has nothing to do with breast cancer yet, right? So maybe this also we can find, we do this drug repositioning. Maybe we can find that this drug also works. Maybe it doesn't work alone for breast cancer, but maybe it works in combination. So there's a lot of room for basically improving, but basically we took basically knowledge from the public databases and curated basically a set of breast cancer agents, and also about a set of 170 combinations that are also available in several resources, basically.
Thank you.
Okay. Thanks.
Ana Paula, please.
Can you hear me?
Yes.
I was wondering if the results from the study, and eventually if it is like improved upon, if they could make an impact into other types of cancer tumors, like from other types of cancer.
Maybe. So then I would just, let's run the same maybe for other tumor, or maybe just let's test the same combinations in others. So what we did is we use this osteosarcoma, is a bone cancer, I think, cell line. So we can have a look. We can see that most of them are or additive, like these ones here, but there's also one combination that is synergistic. So absolutely, I don't know if I want to be successful, maybe I wouldn't just take breast cancer and run it on another tumor without any prior knowledge that maybe this could also work, but it's absolutely possible.
It depends also on the pathway, no? That is following. If the pathway is completely different, you are not blocking.
Exactly. So this is a bit the reasoning behind taking the breast cancer drugs, because they are breast cancer drugs because they are affecting breast cancer related pathways.
Sure.
What about, I mean, in general, many of breast cancer related pathways are MAPK signaling, apoptosis. cell survival and all these things. So they are hitting more or less the same areas, but then there's some tissue differences, and so there's a. So if I would try to use it on another tumor type, I would probably then also try to integrate gene expression data of this tumor type to see, is this pathway actually, is this expressed? Because maybe we have potential crosstalk, but then this pathway is not expressed in liver. So then a potential crosstalk would be inhibited, but it's never actually appearing in the tumor. And then, of course, this is not useful.
Last two questions, Jimena and then Dr. Belmont.
Thank you. First of all, I really like your presentation. And I believe you said that you have taken 10 drug combinations. So my question is, if you have taken other different 10 drug combinations, do you think this will have affected your results, or there will be little alterations, or the behavior of this experiment will remain the same?
Difficult to say, because, so, I mean, we selected the combinations according to the pathway crosstalk index. So we were seeing that, okay, these combinations have potentially a high impact on pathway crosstalk. So that was one measure, right? Then we didn't take the best combinations because the best combinations were already tested or tried in clinical trials or are being used in patients. So there's no point in testing this. Then I think we kind of, so maybe I would expect, if I take 10 combinations with more or less the same pathway crosstalk inhibition values, so they resemble these ones, I would expect the results to be similar, because in the end, we did let's say a targeted selection using the pathway crosstalk inhibition. But then it was me, a PhD student, and my boss that did a kind of like a personal selection of the So it's kind of a randomization of a focused randomization of the combinations that have been selected. So if we would take the same range of pathway crosstalk inhibition scores and do it again, I would expect that the results should be similar.
Thank you.
Dr. Belmont, finally.
Thank you. Give me a second so I can turn on my camera. There you go. Thank you. Dr. Jaeger, am I pronouncing it correctly?
Yeah, very good. Like Jägermeister without Meister.
Thank you, Doctor. Well, my first question would be about the data. How can we access these type of databases, right? Because I think that if we have access to a cross-link inhibition pathway or a drug combination index through a database or repository, right, we as chemists can have ideas of how to enhance or modify the structure of the molecules, right? Something that I really like to do is to modify structures of molecules that are biologically active, and then give like more bio affinities or biodispositions, right, in order to enhance the activity. So my first question would be, do you know if there's a database or repository in which we can look for these pathways or indexes?
So I know the pathway crosstalk inhibition index there's no database. There is a database, I think. I'm hearing myself. So there is the NCI.
I think this is, you have the microphone on, and so I mute you for that. 1 second.
So there's the drug combination database, the DCDB, where we got some drug combinations basically. That could be a good starting point to see which drug combinations are being used, and yeah, what kind of drugs they involve. Otherwise, the pathway cross inhibition index is not, there's no database that basically uses or stores this, because also it depends very much on the pathway definition, right? So I know that biologists, they don't like KEGG pathways so much because they are not complete and they are too messy and whatever. So but but this is what we are having. But if this pathway tomorrow changes because it loses some edges, or it gets some new interactions that are being discovered, then this pathway crosswalk inhibition index, it changes completely, right? So there's no source. I don't know, maybe if now more people also looked into it, and maybe there's some resources, so maybe you can check in the literature if there's some other papers. I think there's like several papers that are looking onto this. But yeah, otherwise the drug combination database could be a starting point for looking at combinations, or the FDA Orange Book that are reporting approved drugs as drug combinations as well. And I think because at that point there was many drug combinations that were in between chemotherapeutics and another very strong drug. So chemotherapeutic goes directly to the DNA. This is something we don't want to improve, right? We want to improve the targeted therapies, because we know that the targeted therapies or precision therapies, they work, but since they are so targeted to one molecule, then we are with the problem is we are running into resistance. And then the idea would be also to see whether we can just switch to another molecule, maybe, or alternate regime between several molecules, or starting directly with the combination, very low level, so we don't run into resistance. We are killing the full tumor, and we don't have this surviving tumor cells that are circulating until they find a new place where they start to proliferate at some point, and a couple of years later, we have tumor again.
Thank you very much. And my next question would be on slide number 22, which I think is like the most popular in the whole presentation. So this is quite interesting. Do you think that in your research community, build up a strategy to transform additivity properties to synergy properties could be something that you would like to explore? Right? Because we already have this information. This information is really important, right? Because we are in a pathway in which we can actually enhance the activity of these molecules, right? If we build up a strategy to modify the structure of these molecules, maybe we can transform, right, the additivity towards synergy, right?
Yeah.
So my question would be, do you have any idea which strategy would be the best to follow in those terms? Meaning which properties, specifically for this study, right? Which properties of these molecules would you think, in your experience testing them, can enhance that property?
Well, in the end, it's about binding, right? I mean, you're binding it to a target, and you're inhibiting this target. So this target cannot propagate information, basically. So this is basically kind of the key of the pathway crosstalk. We want to inhibit the pathway crosstalk. So it's about maybe a better binding to that protein, better binding affinity, tighter binding, maybe also using instead of 2 drugs, maybe we can use 3, right? And we can, so we are not only touching 2 points in the pathway, but we are touching 3. So one above and one below, for instance, or try to find also where can we inhibit at different layers, right? So usually on top is the receptor, but then there's so much going on in the cell until it reaches the nucleus. So can we make sure that we are inhibiting it in the nucleus, in the middle layer, and in the receptor area, for instance? So maybe this would be something that I would explore. Then from a chemistry's point of view, I mean, better drugs will be better combinatorial drugs as well, right? So
I think that the main problem is that you are using drugs that are already known. Entering inside modification of the drug means entering inside a drug discovery process since the beginning. So maybe the knowledge that have been acquired in these crosstalks process pathways can help in enhancing the activity, but means starting a completely new drug discovery process.
Absolutely, and
so it means a new word, basically.
Even if you are having, for instance, so now this drug combination it is approved for breast cancer, and the raloxifene is one of the most famous, and then the cabozantinib is, I don't know if it's by now approved for another drug type. But so if we would now want to move to the clinics, we would again start a completely new trial. So we cannot use the information that, hey, raloxifene is already tested and approved and it's safe, and anyway we are using less concentration, so it should be safe, right? But we have to start again from the beginning. And then, for instance, in this case, it's different companies. They are like, the drugs are already patented. So basically it's a little bit a lost battle for a small research group to push. I mean, we try to explore a little bit what we can do with this drug combination, if we can push it a little bit further to the clinics, but it was not really in our hands, or maybe we should, no, I mean, too difficult.
Yeah. I think we need to start closing up. Dr. Belmont, do you have another?
Yes, just one more, because this is amazing and I don't want to lose my chance. In the last slide, you talk about inhibition of the BCL2 pathway, right?
A molecule, yeah.
Yes, this one exactly. I am really interested in that pathway. Right? I have a special interest in that pathway. Do you know which is like the meta in research about the combinatory inhibition for the BCL2 family proteins?
No idea. This is no. I'm a bit like a large scale person, right? So I'm doing everything at large scale, and then now we were looking, in this case, we were looking into this. Okay, what is actually raloxifene doing? What is actually cabozantinib doing? So let's say this is a bit by coincidence. It's not coincident that BCL2 is here, but we didn't really look. We looked at the drug targets, and then we looked, okay, what is being shared? But I have no knowledge on the BCL2 proteins, or neither on everything that is around here. Right? It happens to be in this crosstalk area, and then I guess also it's a protein that it can be easily here tested in Western blot, and so that's like common marker or common antibodies that are available. So we pick the ones that we can easily test in a Western blot.
Thank you very much, Doctor. Again, congratulations. These are the kind of works that I think change the world. Please don't stop.
Thank you.
So thank you very much, Dr. Jaeger. Really appreciate your time and being available. And I think it's almost time that you finished work today. So it's about time you go back home and enjoy your evening. And Professor Gonzalez, do you want to say something about? Yeah.
Yes, of course. Also, it's also always happy to hear from you, Samira. The previous time with us, I learned a lot, and this time I learned even more. So I hope that is just the beginnings of so many other interventions that we can have. And I'm going to show you.
Stop sharing.
Oh, I closed it. Yes, please. I'm going to show you. I'll just display your certificate of participation that we made.
You are collecting them from our university.
Yeah, I know.
I will put them here in the wall.
Yes, please. When you get 5, you get an extra flight ticket to come to Mexico.
Oh, don't promise too much.
Don't say too much.
So I said 5.
Well, I'm already
Thank you very
halfway.
much for joining. Thank you very much for being with us.
Thank you for giving the opportunity.
One of the talks that got the more audience. There were like 60 people connected here, and we also had another bunch of people watching Facebook Live. So this is very good. And well, thank you very much, and we'll stay in touch.
Absolutely. Thank you for the opportunity, and enjoy the rest of the STEM week.
Thank you.
Thank you very much. I'm just gonna give some announcements for the next talk. We had a biotechnology entrepreneurship talk, but it has to be rescheduled. Our speaker had a trouble a bit with timing, so I think we'll have to leave that talk for later. Not during this STEM week, but I'll let you know when we postpone. Okay, it's not canceled. I don't want to say that it will not be canceled. It's just that we cannot have it today at 12, but we will have it soon I hope, and I'll let you know about the time. But we do have the other one at 3 p.m. Let me just open the schedule very quickly here. Well, the one from UNAM and the physics. You can, I think you can see my screen, so that's good. The one at 3 p.m. we will do have it. Okay, so stay tuned, join, and I'll see you around. Okay, so there is no talk at 12. Keep it in mind.
Thank you very much. Thank you, Samira.
Bye-bye.
Thank you.
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 Thank you.